Hello and welcome to today's video. If you are a biology student or simply curious about the wonders of science, you are in the right place. Today, we will look at 3 fascinating biology practical questions. Question 1 will explore the effects of osmosis on plant strips. Question 2 will examine the structure and function of a lung specimen. And question 3 will uncover the anatomy of an onion bulb. Grab your rough coat and let's start learning biology fun and engaging. In KCSE 1990 Biological Sciences Paper 2, the practical requirements for an examination which was taking 1.75 of an hour were as follows. Question 1. Question 1, a raw power labeled specimen Q. Question 2, a piece of lung labeled specimen R. And question 3 was an onion bulb labeled specimen C. The question 1 questions were as follows. You are provided with a specimen labeled Q. Part A. Giving a reason, suggest the method of seed dispersal in this specimen. You are given 2 marks, one for method, the other one for reason. In question 1b, you are told to remove the seeds and fresh part from the specimen, leaving a peeling of about 3 millimeters thick. From the peeling, cut a piece measuring about 5 centimeters long along the length of the peeling and 1 centimeter wide. Obtain 3 equal strips from the piece by cutting along its length. Immerse 1 strip in distilled water, another in 1% sodium chloride solution, and the remaining one in 1% sucrose solution. Leave the setup for at least 30 minutes, after which you remove and compare the shape and flexibility of the strips. Record your observations in the table below. You get 6 marks. Strip immersed in shape, flexibility, distilled water, 1% sodium chloride solution, and 1% sucrose solution. Part C was you account for the shape of the strips at the end of the experiment after being immersed in distilled water. Get 3 marks. And then after being immersed in 1% sodium chloride solution, you get 2 marks. Then in D, you are asked how did the concentration of the cell sap in the specimen at the beginning of the experiment compare to that of 1% sucrose solution. 1 mark. B, Roman 3. What happens to the specimen when released after squeezing under water? So I think there was a mix-up there. So question 1 was reaching D. So you have A, B, C, and D for question 1. So let's go to question 2. A slight mix-up. The mouse disorganized us. Question 2. You are provided with specimen R. Using a hand lens, examine the specimen. Describe the observable features of this specimen, 3 marks. Immerse the specimen in water, record your observation, and then squeeze the specimen out of water and you record your observations. B. Roman 3. What happens to the specimen when released after squeezing underwater? C. Roman 1. Name the organ from which specimen R was obtained, a mark. Give a reason for your answer. In C1 above, a mark. Observe the specimen while in water. Make a labeled plan diagram of the specimen. 5 marks. And then from your observation, explain how the organ from which this specimen was obtained is adapting to perform its function. 2 marks. Question 3. You are provided with the specimen labeled S. What is the name given to this type of plant structure? And in B, using a sharp blade, make a longitudinal section of the specimen to obtain 2 halves or equal halves, and then you make a ribbon drawing of one of the sections. From observable features only, explain how specimen is adapted to its function. So specimen for question 1 is there, a raw pow. And these were the responses according to the council marking scheme. The method is animal dispersal, or you can say man or birds, but you should reject insects. Insects cannot disperse purple. Reason: it is succulent, fleshy, edible, slimy seed coat, and digestible seed. Any of those, you get a mark. But for a structured answer, the red one just gives you the same thing, but structured. Animal dispersal. Reason: the pawpaw fruit is fresh, sweet, and brightly colored, making it attractive to animals that eat the fruit and disperse the seeds through their droppings. 1B. Remove the seeds and fresh part from the specimen, leaving a peeling of about 2 millimeters. Leaving a peeling of 3 millimeters thick. From the peeling, cut a piece measuring about 5 centimeters long along the length of the peeling and 1 centimeter wide. Obtain 3 equal strips from the piece by cutting along its length. Immerse 1 strip in distilled water, another in 1% sodium chloride solution, and the remaining one in 1% sucrose solution. Leave the setup for at least 30 minutes, after which you remove and compare the shape and flexibility of the strips. The marking scheme, the examination council marking scheme, when you immerse in distilled water, the strip is carved outwards, and you say not flexible, or you can say not turgid, or carved towards the peel, or carved towards the green side. All those answers are correct. In 1% sodium chloride solution, it is carved inwards, or carved towards the fresh part, and the flexibility, it is flexible, not firm, it is flaccid, it is so soft, or once to that effect. 1% sucrose solution, it is slightly carved outwards. Alright, slightly carved towards the peel, green part, or green side. Slightly flexible. As simple as that. You could have accepted the shapes. For the shapes, you could have accepted the drawings given here. In distilled water, you can see the shape of the drawing. In 1% sodium chloride, you can see the shape, and in 1% sucrose solution. In distilled water, we account for the results. Account for the shape of the strips at the end of the experiment after being immersed in distilled water. The inner cells absorb more water than the outer cells by osmosis. 2 ticks up to there. The cuticle is impervious to water, a tick, and hence cuticle resists stretching, hence curving outwards, a tick. The cell sap is hypotonic to distilled water, a tick, or the converse. The distilled water is hypotonic to the cell sap. 5 marks, maximum 3. A structured way of doing it is that the cells become turgid as water moves into them, causing the strip to swell and become firm and less flexible. In distilled water, the water potential is higher than that of the pulp cells. As a result, water enters the cells by osmosis, causing them to become turgid. When cells become turgid, they swell and their rigid cell walls resist further expansion. This makes the overall tissues less flexible because the cells are firm and cannot bend easily. Continuation of the marking scheme, Cancer marking scheme. In 1% sodium chloride solution, you state as follows: the inner cells lose more water than the outer cells. You can also read the inner side loses more water than the outer side. That one can also work. By osmosis, the cuticle is impervious to water. Up to there we have 3 marks because I told you as in colon is a mark. The cell sap is hypotonic to 1% sodium chloride solution, a tick. The cuticle resists stretching. The strip becomes flaccid, or the strip is plasmolyzed, or the strip shrinks, a tick. 5 marks, maximum 2 marks. You can also put it this way. In this solution, the water potential is closer to that of the cell sap, so there is minimal water movement. This causes the strip to retain some flexibility as the cells are neither fully turgid nor plasmolyzed. The term flaccid is biologically accurate here. For the 1% sucrose solution, when we say it is slightly flexible, it's reasonable because the cells lose water to the sucrose solution due to osmosis, becoming plasmolyzed. As a result, the strip shrinks and becomes less firm, but may retain a slight flexibility depending on the degree of plasmolysis. This is something you have to note. In C1 and 2, marks for osmosis should be awarded once. If awarded in 1, then should not be awarded in 2. That is the word osmosis. Proper or correct definition of osmosis should be awarded marks once, if the word osmosis is not mentioned. Cell sap was hypotonic to 1% sucrose solution, or the converse, that 1% sucrose solution was hypotonic to cell sap. That is the response of D, which was 1 mark. For question 2, those are the specimens. You can see the piece of lung, and you can see the lungs on the other side. All right, there you are. Describe the observable features of this specimen. And you can see brand vessels or capillaries or arteries, arterioles, or veins. You can also see hair holes, vessels, passages, cavities, sacs, alveoli, branches, all those ones, any one of those ones you can mention. And then you can see it as a thin membrane or lining on the outer side of the specimen. But you can also say that the specimen has a spongy texture, or you say it is pinkish or grayish in color. The surface is uneven with visible air pockets or tiny holes. Reject if somebody talks about pulmonary artery and vein or aorta or bronchus. They cannot be seen. So the council marking scheme is the black. The red is just addition. Immerse the specimen in water record your observation. The observation is that there are air bubbles, or you can say the specimen floats on water. Reject water bubbles or foam. Roman 2, squeeze the specimen underwater, record your observation. More air bubbles is the observation, or you just say effervescence. Or you can say air bubbles are released from the specimen. Roman 3, what happens to the specimen when released after squeezing underwater? It expands, or it soaks water, or it absorbs water, or regains shape, size, or volume. And the second point is that it floats on water surface. It is less dense than water, so that you get your 2 marks. And the structure, the other way is that the specimen returns to its original shape due to its elastic nature. It rises back to the surface of the water, indicating buoyancy. Name the organ from which air was obtained. It is the lung. Give a reason for your answer in C1 above. It is a spongy, or it has hair sacs, it has capillaries, spaces, passages, bronchus, or alveoli. Or you can just say the spongy texture and the presence of air pockets or alveoli suggest it is adapted for gaseous exchange. Observe the specimen while in water. Make a rebel plan diagram of the specimen, 5 marks. The marking scheme is there. You can see we are marking tissue of alveoli. Reject alveoli alone. We are marking pleural membrane. This is a plan diagram, meaning there are some hypothetical instincts. You have the air sac, you have the bronchiole, you have the air space, blood vessel or capillary, artery, arterial, venue or veins. Note, since the outline was variable, marks were awarded for correct labels only. So we were not marking for the drawing. After all, it was a plan. Reject pulmonary artery, vein, aorta. They cannot be seen. Deny marks from bronchus or bronchus because they are indicated. From your observations, explain how the organ from which the specimen was obtained is adapted to perform its function. 2 marks, marking scheme. Air spaces or sacs, holes to increase surface area for gaseous exchange. Bronchus for distribution of gases to and from the lung. Thin lining for protection, tissues are airtight. Blood vessels or capillaries for transport or facilitating gaseous exchange. Or you can just say, the lung has a spongy texture with numerous air spaces, alveoli, to provide a large surface area for gaseous exchange. Its elasticity allows it to expand and contract during breathing. Notice that, as usual, the black is the council marking scheme, the red is my own addition. 4 marks, maximum 2. Marks only awarded for correct observation and function. Question 3, specimen, an onion bulb, and how it was cut, half of it. Marking scheme. What is the name given to this type of branch structure? It's a bulb. Or you say onion bulb or bulb onion. But you reject onion, all right? Aroon or onion plant or onion bud or bulb onion. You reject those ones. Bulb onion, should reject it. And that is the diagram. Using a sharp brain, make a longitudinal section of specimen to obtain 2 equal halves. You have seen them. Make a ribbon drawing of one of the sections. You get 5 marks. And you can see where the marks were being put. Mark for correct shape. Mark in the stem. Mark for continuous outline. All right. Rebels, fresh leaf, terminal bud, scale leaf, adventitious roots. This is the way it was being marked. For diagram marks, things to look for: shape, bulbing, fresh leaves must be crossed, leaves originate from stem. Outline, smooth, scale leaf must be continuous. Shape of stem should be crossed, dome-shaped, not shaded. Award mark even dotted. Inside is dotted. Rebels. Rebel lines must touch the structure to be rebelled. Each correct rebel is half a mark. Each correct rebel is half a mark. Award mark for root if it arises from the stem. From observable features only, explain how the specimen is adapted to its function. Roots, absorption of water and mineral salts or anchorage. Fresh leaves for storage of food. Stem for attachment of buds or leaves. Auxiliary or terminal bud for vegetative propagation or development of a new plant or shoot. Reject development of new leaves. Adaptation for the onion bulb to its function. This one is now mine, the structured way of looking at it. So 1, fresh scale leaves store food or starch and sugars and water, enabling the plant to survive unfavorable conditions like drought. Dry outer scales provide mechanical protection and reduce water loss by being impermeable. Basal plate anchors the bulb to the soil and supports the development of roots. Adventitious roots facilitate efficient absorption of water and nutrients, and apical bud ensures the bulb can sprout into a new plant when conditions are favorable. 4 marks, 1 mark for each correctly explained observable feature. This is my own way of looking at the adaptations, but the council ones are in black. Closing remarks. Thanks you so much for watching. I hope you have enjoyed exploring this exciting biology practical and gained a clear understanding of osmosis, lung structure, and onion bulb anatomy. Don't forget to try these activities yourself and share your thoughts or questions in the comments below. If you found this video helpful, please give it a thumbs up, share it with your friends, and subscribe for more educational content. Until next time, stay curious, and keep exploring the wonders of biology. See you in the next video. And we acknowledge, before we wrap up, ChatGPT assisting in structuring the opening and closing remarks, title, description, hashtags, and clarifying the concepts in the topic. The student who inspired me to come with this practical. The diagrams and photographs here used here were sourced from my individual work. Finally, the slides were designed by me, and I thank everybody who directly or indirectly contributed to making of this presentation, like the butcherman who was able to give me the lung and the piece of the lung. All right, so that ends our video, and we see you next time.